we'll be discussing current regulatory issues that we see in drug product manufacturing. And so this will be focused on the biological license applications. So moving forward, this is my presentation outline. So I will discuss the content of the BLA submissions as guided by the documents and regulations. Also, I will discuss common deficiencies that we see related to process validation in biological license applications. And I'll focus on 3 areas: sterilizing filtration, post-reconstitution, and post-dilution storage, as well as container closure integrity. At the end, I'll draw some conclusions, and at the end of this talk, there are some reference slides for you all to review on your own time. So first, I'd like to begin all of my presentations with a review of the laws and regulations that we use to govern and to guide our BLA application reviews. So the first regulation or law that I'll discuss is the Public Health Service Act, which basically says that biological products must be safe, pure, and potent. Also, it says that the facility in which the biological product is manufactured must also, that the biological product is manufactured, processed, packed, or held must meet standards designed to assure that the biological product continues to be safe, pure, and potent. In addition, the Federal Food, Drug, and Cosmetic Act interprets that biological products are also drugs. Okay? So both of these laws will give us the authority to conduct inspections. And so we conduct inspections with these laws in mind. Okay, so both the PHS and FD&C Acts require that biological products must be manufactured under current good manufacturing practices as described in the Code of Federal Regulations, Sections 210 and 211, as well as 600 through 680. So moving forward, we also look at the Code of Federal Regulations that I just mentioned that discusses the control of microbiological contamination. It says that appropriate written procedures designed to prevent microbial contamination of drug products purporting to be sterile shall be established and followed. Such procedures shall include validation of all aseptic and sterilization processes. This addresses the validation of aseptic and sterilization processes. And you can refer to the Code of Federal Regulations, Part 211, for more regulations applicable to sterile drug products. And so this is the basis for all of our reviews. Moving on, we also refer to guidances that are also available to you. I think Marla she mentioned these guidances as well in her talk, so I might not go into much more detail here, but basically we consider the guidance document titled Submission of Documentation for Sterilization, Process Validation, and Applications for Human and Veterinary Drug Products. And so this guidance document describes sterilization process validation information that needs to be included in your BLAs. Secondly, the sterile drug products produced by aseptic processing, current good manufacturing practice. This will give you guidance on how to comply with the CGMP regulations. And this is to be used in conjunction with other compliance programs and guidance documents. Also, the container closure system integrity testing in lieu of sterility testing as a component of the stability protocol for sterile products. I will go into this guidance document in a bit more detail in the context of a case study and also some common deficiencies. And there's another draft guidance that discusses reportable CMC changes for approved drug and biologics products. Okay, so moving forward, I'm going to go into some common deficiencies that we see when we're reviewing BLAs. And so as I stated before, I'll be discussing 3 major topics today. There are many more, but I wanted to focus in on some of the ones that are most critical. So the first one is sterilizing filtration. This is critical for a BLA product because typically the products that we see cannot be terminally sterilized. So the sterilizing filter and the sterilizing filtration process must be adequately validated, adequately described in the BLA so that we can be sure that the process is producing a sterile product at the end. And so we refer to the PDA Technical Report 26, which discusses sterilizing filtration of liquids for general guidance. So I'm going to go into a little more detail on this and discuss integrity testing, which is the area that we see a lot of deficiencies, as well as a description of the process parameters in the microbial retention validation studies. I will also go through a case study as an example. So common deficiencies as it relates to sterilizing filter integrity testing. We often see BLAs that provide no information or insufficient information for product bubble point determination. We often see that the test description is missing or insufficient. We often see acceptance criteria listed only as pass. We like to see a numerical value for bubble point or filter integrity test results. In addition, applicants will often leave out the wetting agent used in the bubble point test. And quite often, the sterilizing filter integrity test results from process validation lots is not provided. So when these things are missing or insufficient in the BLA, we often have to send an information request requesting this information. In addition, many times BLAs are missing a filtration time limit or product contact time. The time limit is not included in the parameters, or the proposed time limit is significantly longer than what is required for the production process and is not appropriately validated by the microbial retention study. So again, we would have to request this information from the applicant or request that the studies be done repeated in order to address these deficiencies. Most recently, we've been seeing a lot of pressure or flow rate limits related deficiencies, where peristaltic pump speed range is given in lieu of pressure or flow rate limit for sterilizing filtration. Pump speed is variable. Pump speed can change depending on what pump is connected to the system. Pump speed should be correlated to a process parameter that is validated by the microbial retention study, in which case we would like to see flow rate or pressure. Also, controls also need to be in place to ensure that the pressure or flow rate limit that is validated by the microbial retention study is not exceeded during production. So all of this information has to be included in the BLA. Most often, we also see retention study reports and or viability data that's not provided in addition to the summary data, or the study report was not legible. Many times we see study reports that have been scanned and illegible. They may not be in English. These also can hold up our review because we have to request the summary data, and also the viability data is not included. We have to see a control that shows that your challenge organism is viable in your study. Also, scaled down study parameters were not compared to production parameters in your microbial retention validation studies, or the scaled down study did not support the worst case production parameters. So in which case we need to see these parameters such as product contact time, flow rate or pressure, product volume, per unit of membrane surface area, temperature. All of those parameters have to be addressed in the scaled down study. In addition, that we see quite often inadequate justification for not performing the study as a single stage direct challenge with unmodified product under worst case conditions. So we usually see this when the drug product formulation is bactericidal, and so water is used as a surrogate. Also, we see a study design modified to accommodate an unnecessarily long filtration time limit. Okay, so here's a case study where a microbial retention study was completed as a two-stage test. which means that there's product conditioning where the drug product is pushed through the sterilizing filter, and then followed by the bacterial challenge, where the challenge organism is suspended in water because of the bactericidal properties of the drug product. However, water is not a suitable surrogate solution for many BLA products, in particular this one in the case study. And also studies were not performed to identify what part of the drug product formulation was bactericidal. And so that would allow for a more suitable study design. Many times if you identify what is killing the bacteria, you can remove it, and the other components of the drug product formulation can remain in order to resemble the drug product more closely. So as a result, the following post-marketing commitment was agreed upon, and I'll read it word for word. The microbial retention study was done with purified water as a surrogate solution for the drug product. Perform a repeat microbial retention study for the sterilizing filter using a suitable surrogate solution. Product attributes of the surrogate solution that are known to affect microbial retention, such as surface tension, viscosity, ionic strength, etc., should model the drug product as closely as possible while preserving viability of the challenge organism. Alternatively, a reduced exposure time approach may be appropriate. Next, I'd like to discuss another very important issue that we see upon BLA review, and this has to do with post-reconstitution and post-solution storage. We often see lyophilized products that must be reconstituted prior to administration, and this is directed by the label in the instructions. Proposed post-reconstitution storage time has to be supported by microbial challenge studies in order to demonstrate that the product does not support microbial growth under the proposed storage conditions. This also applies to post-dilution storage time for liquid products. So, in more detail, challenge studies must be conducted using a panel of microorganisms provided by the USP, and also include a typical skin flora or species associated with hospital-borne infections. The challenge level should be less than 100 colony-forming units per ml. The temperatures described in the proposed product's labeling should also be tested. Also, test duration should be twice the recommended storage period and use the label recommended diluent. And also, an increase from the initial count has to be defined as less than half a log unit higher than the initial inoculum. So here's a case study to put this all in perspective. Product A has to be diluted in 0.9% saline prior to administration. On the label, the post-dilution storage conditions state that it is stable for up to 24 hours at 2 to 8 degrees Celsius, or up to 12 hours at room temperature. The applicant provided a growth promotion study result that showed that the drug product was growth promoting for Pseudomonas aeruginosa by 32 hours at 2 to 8 degrees Celsius, and also by 24 hours at room temperature. The results also show that the drug product was growth promoting for E. coli by 16 hours at room temperature, which is the results show a twofold increase in CFU at the 12-hour time point in duplicate samples. Therefore, the label was revised to remove the room temperature storage time. Moving forward to container closure integrity. So we are guided by the 1994 guidance that states that the sterility testing at the initial time point is not considered sufficient to demonstrate the microbial integrity of a container closure system. Documentation of the sensitivity of the container closure integrity test should be provided. In addition, the 2008 guidance states that sterility tests are not recommended as a component of a stability program for confirming the continued sterility throughout a product's shelf life or dating period. Alternatives to sterility testing might include any properly validated or chemical container enclosure system integrity tests or microbiological container enclosure system integrity tests. For example, microbial challenge or immersion tests. Also, a test method is adequately validated if it has been proven through scientifically accepted studies to be capable of detecting a breach in container enclosure system integrity. An appropriate container enclosure system integrity test should be conducted annually and at expiration or as otherwise required by applicable regulations. So, we still see these common deficiencies. The container closure integrity test was not included in the stability program. We also see inadequate qualification of the container closure integrity test, which includes inadequate description of the test method, which does not include a sensitivity of the method. The sensitivity of the method either is not known or described. And also we see a lack of appropriate controls. Also, vial capping parameters are not described in the BLA, and this has been shown to have an impact on container closure. So that information also needs to be included in the BLA. In addition, container closure integrity of syringes, particularly during shipping, studies have shown that due to air pressure in air transportation of syringes can lead to plunger movement, which can affect the container closure integrity. Those studies have to be done adequately and described fully in the BLA. So here's an example of a deficiency that we've seen where the container closure integrity test had an inadequate positive control. So an applicant proposed to use a container closure integrity test that was capable of detecting a defect as small as 160 microns. So the positive control that they used during the method validation was a container prepared with a 160 micron defect. Current CCIT methods are capable of detecting leaks of less than or equal to 20 microns, according to the PDA technical document I mentioned earlier. So system suitability controls with a smaller defect size should be used for routine testing. So our resolution, we sent the following information request to the applicant, which states, the system suitability controls for the container closure integrity testing syringes and pens are prepared with a relatively large defect size. System suitability controls with a smaller defect size should be used for routine testing. The study performed by the contract lab showed that the method is capable of detecting 5, 10, and 30 micron defects. As a result, the applicant did commit to implementing a system suitability control with a smaller defect size, in which case they used less than or equal to 20 microns. In conclusion, sterilizing filtration described in the BLA should include the integrity testing information. Filtration parameters should be supported by the microbial retention study. Modifications to the microbial retention study should be made only when necessary and should be supported by viability study data. Post-reconstitution and post-dilution storage conditions have to be supported by growth promotion study data. Also, the container closure integrity test has to be used in lieu of sterility for drug products and has to be tested annually through expiry. CCIT method validation studies should demonstrate adequate sensitivity using the appropriate controls. Please refer to the guidance documents and pre-meeting comments for the drug product information that should be included in your BLA. And also, this is critical, in conclusion, the FDA review timelines are based on the expectation that applications are complete at the time of submission. Any changes to that or any missing information can prolong the review process and make it more difficult. So it's better to have all the information included in your BLA upon submission. So with that, I'd like to make an acknowledgement to Dr. Lynn Enzor, the Director of our division, my branch chief, Patricia Hughes, and also a colleague, Colleen Thomas, for help with this presentation. Thank you very much.